Hello student, in our today's lecture we are going to start with the next point from the topic, that is the acquired or adaptive immunity. As you know, this adaptive or acquired immunity is also called as specific immunity. It is also called as a specific immunity. Okay. This specific immunity is depend on how many antigen it depends on. It is called as adaptive immunity because of it depends on the chances of antigen for lifetime, and according to that we are developing or we are forming antibodies against that, right? So especially related with the acquired immunity which is acquired during our lifetime, and that is especially depend on how the chances of antigen invades to us. Okay, it depends totally on that, how many antigens invade to that person, and then only the body produces the antibodies. So it is called as adaptive immunity, and it is very specific to its antigenic invasion. Clear? Antibody production is very specific to its antigenic invasion. So because of that, it is called as a specific immunity. Clear, everyone? So this acquired immunity having some features. So what are the features of acquired immunity? So the next one is the characteristic features of acquired immunity. Acquired immunity. We have studied the difference between the innate and acquired immunity, as you know. The first, now we are turning towards the features of acquired immunity. The first feature of the acquired immunity is the specificity. Specificity, right? As you know, it's named these as a specific immunity. So, antibodies production is very specific, is very specific to its antigenic invasion. All right, that means which type of antigen invades, whether it is a virus or bacteria. There are so many viruses are there, so it depends on which type of antigen invades in a body, and according to our body produces the antibodies, right? So it comes under the specificity. The second feature is a diversity. Okay. So as you know, the one of the characteristic feature of acquired immunity is, especially there is a tremendous diversity is in the form, as well as there are different types of pathogens or the bacteria as well as viruses. There is a numerous number of bacteria, viruses are there, and according to that, for every different virus or bacteria, different types of antibodies are generated. That means nowadays our body having number of antibodies. That means there is tremendous diversity is in the antibodies also. Okay. So there are there is tremendous diversity is in the antibodies. So we have numerous antibody. So as there are numerous antigens, and against this antigen, our body having numerous antibodies according to this antigens, right? So there is a tremendous diversity is in the antibodies also, right? So this is the another one, another feature. Then the next one is a discrimination between self and non-self particle. Third characteristic, it is very important one, discrimination between self and non-self particle. So discrimination means distinguish. Okay, it means distinguish. So one of the feature of acquired or adaptive immunity is the discrimination between self and non-self particle. That means the innate immunity, when it has to work, it works at that time, it has to identify that pathogen, right, among the self particles. That means whatever the foreign material which enters in our body, that must be recognized from the self particle. That means the non-self particle must be identified. Okay, for example, these are different types of material is entering in our body. Among this is the only a non-self particle and this is the self particle. For example, we are eating the food. When we are eating the food, during that, if that food is contaminated, okay, that time there are so many micromolecules which are formed. Okay, these molecules are useful to our body. These are the nutrients, right? Among that nutrient, which are the non-self particle and which are the foreign material which is causing disease, that must be identified. So it is a very important feature of acquired immunity. As we have defined the immunity, that is, the immunity is the ability of the body to identify, right? First of all, to identify, then to destroy, and then to eliminate, right? So first of all, identification is very must from the self particles. So many substances which are entering in our body or in our bloodstream. So many proteins are, as we have studied, so many proteins are present in our body, in a bloodstream, they are flowing, and among that, whatever the material which enters, the bacteria or virus which enters in our body, that is also a proteinous in nature. So among that, useful protein, which is a harmful one, that means which is a bacterial one, that must be identified. Or what is the toxic material, that must be identified, and that is called as a discrimination between self and non-self particle. When it discriminates or separates out the non-self or foreign particle, and then only our cells, the phagocytic cells or the immune cells, attacks on that pathogen or that toxic substance to remove it, right? So this is, one of the important function, and very, very important function which is important to form a vaccine, that is a memory. So what is this memory? So related with the memory, when the antigen invades to our body, at that time, as you know, this is the first encounter of that antigen. That means at very first time that antigen invades to our body, right? At that time, this is our first encounter. If during this first encounter, the amount of antigen is very, very less against this antigen, but this antigen get familiar with our body, and to respond to this antigen, our body produces the antibodies. So this is the first encounter. That means this antigen invades in our body, and against this antigen, our body has produced antibodies. Clear? This is the first encounter. Sometimes this first encounter may lead us into the disease stage. Okay, in a disease stage. and we get suffered with that particular disease, right? But the feature of acquired immunity is that it keeps in a memory. It keeps it in a memory that this particular antigen has previously visited to our body or entered in our body. Clear? That means this antigenic invasion is kept into the mind. There are memory cells which are present in our body, and these are keeping it in their memory. And when again at the second encounter, when this antigen tries to invade our body at that time, but we have antibodies against it, so this antibodies works on it and destroy that antigen. That means the first encounter which is kept into the mind that this antigen was previously visited and leads us into the disease stage, or such an antigen, such a pathogen, or such a virus was invented in my body, and my body having number of antibodies. So this is the very unique feature of acquired immunity, that is a memory. And nowadays we are using this feature of acquired immunity for the vaccine production. This is very important one for vaccine production. Okay, so during this vaccine production, what we are doing, we are just entering the antigen by forcefully entering the antigen. Weakened or dead form antigen is introduced in our body, and we are just stimulating our immune system to produce the antibodies. So whatever the immunization plan run up to the age of 6 in our by our government, that means up to the age of 6, our body has produced number of antibodies against several diseases, right? So when occasionally or by during our lifetime, if that virus enters in my body, but my body already has prepared antibodies against that, so that antibodies works on that antigen, and I will not get suffered with that disease. Okay, that was just a feature of this acquired immunity, and that is very important feature. That's a memory. Clear, everyone? So these are the 4 features of acquired immunity. Can be asked for 2 marks. The first one is the specificity. Listen carefully, the sentence. Okay, antibodies production is very, very specific to its antigenic invasion. That is the specificity. Which type of antigen invades, and according to that the antibodies are produced, right? Then diversity. As there is numerous varieties of antigens, okay, so to prevent that antigen, our body also having different types of, there is tremendous diversity is in the antibodies also. Clear? That is the diversity. So discrimination between self and non-self particle. That means to identify non-self particle from the self particle. That is very unique one, so that when it identifies that non-self particle, then and then only the phagocytic cell or the immune cell work on that, right? Or the antibodies works on that. Then the next one is a memory. Memory is the feature, very important feature, which is used for vaccine production. That is the first encounter is kept into the mind, okay, into the memory, especially it is kept into the memory, and our body is familiar with that antigen, and so that our body has produced already having the antibodies against that, yes, antigen. So that is the next one, right? Now we are turning towards the types of acquired immunity. So this question is asked for 2 marks. So prepare for 2 marks. The first one is the specificity. Second one is the, yes, everyone, here, Second one is the diversity. Third one is the discrimination between self and non-self particle, and the fourth one is the memory. This is the next one, types of acquired immunity. So, acquired immunity is typed into two. It is typed into two. First one is the active immunity, active acquired immunity, and a passive acquired immunity. So, active acquired immunity and the second one is a passive acquired immunity. So, active acquired is further grouped into two: natural and artificial. So, how we name natural active acquired immunity? Then the next one is the artificial. Artificial active acquired immunity. Then this passive is again grouped into two. It is also grouped into two. The first one is natural, but natural passive acquired immunity, and the next one is the artificial. Artificial passive acquired immunity. So the immunity is divided into, the acquired immunity is divided into active immunity and a passive immunity. Very important one is a active immunity and another one is a passive acquired immunity. So what do you mean by active? Active means active immunity. Active acquired immunity. Active means what? When any antigen invades to our body, our body produces antibodies against that antigen. That means our body is actively participating for antibodies production. Right? So active acquired immunity, especially when we are saying active, that means our body is participating in production of antibodies. Keep in mind, active means our body is participating in production of antibodies. That means we are actively participating in a production of antibodies. While passive means what? We are not involved in a passive immunity. Our body is not involved. We are taken ready-made form of antibodies. That means passive means the antibodies are produced by another organism and we are taken in as a ready-made form. Okay, ready-made form. Okay, ready-made antibodies are taken. That is the passive antibodies. We are actively ourselves are participating in a production of antibodies, then it is called as a active immunity, active acquired immunity, in which we are participating in a production of antibodies, while in case of passive, the antibodies are produced by another individual and we are taking it into the ready-made form. We are just picking that antibodies and inserting in our body. That is simply called as a passive immunity. Clear, everyone? Active and passive, clear? Okay, very clear to everyone. Okay, now we are running towards the next point. That is, the active as well as passive immunity is divided into natural and artificial. Natural and artificial. So, the first one, natural active acquired immunity. That means when any antigen invades to our body, obviously our body produces antibodies against it. That is the natural one, right? That is the natural one. When any antigen enters in our body produces the antibodies against it. For example is a chickenpox, measles. Okay. Example, when chickenpox enters, chickenpox and measles. Measles, right? So in our chickenpox and measles, this antigen when invades in our body produces the antibodies against it. Okay? So this is the natural. By natural way, when any antigen invades in our body produces antibodies. That's it. Okay, that is a natural acquired immunity. So artificial acquired immunity. Artificial acquired immunity, that means our body is producing antibodies, right? But it is by artificial way. That means we are forcefully injecting the antigen in our body, and our body itself produces the antibodies because it's the active immunity, right? And this is especially practiced in a vaccination. This is in a vaccination or immunization plan. Okay, during vaccination, what we are doing? We are injecting dead form, or we are injecting dead or very weak antigen forcefully in our body, and our body produces the antibodies against it. That is the artificial active immunity, acquired immunity, right? That means artificially, by artificial way, we are injecting the antigen in a body, and so that our body produces the antibodies, right? So this is the artificial way. Okay, then the next one, passive acquired immunity. It is divided into natural and artificial immunity. Passive means we are just taking ready-made antibodies from another person. By natural way, from whom we are getting the antibodies by natural way? Yes, that is our mother. Okay, so from our mother to her fetus, mother to fetus, via transplacental transmission, via placenta. When baby is in the womb of mother, at that time the antibodies cross via placenta enters into the fetus, right? Ready-made antibodies taken from mother to the fetus. Then another one, after birth. After birth, the first milk emitted from the mammary gland of the mother is called as, yes, we have studied in a previous topic. Yes, it is called as a colostrum. Okay, we have studied it, colostrum. Colostrum. This colostrum is a source of antibodies, richest source of antibodies, and so that we are awareing people to feed the colostrum to their babies, so that they will get the antibodies, which are ready-made antibodies from mother to her child, right? That means when the fetus is in the womb of mother, during that, via the placenta, the antibodies are taken from the mother's blood to the fetus, right? And after birth, the colostrum is the richest source of antibodies, and that is given to the babies, right? So, which is a thick yellowish color milk which is emitted from the mammary glands of the mother. That is very important for the fetal growth. Even though we are also consuming the cow colostrum, domestic animals colostrum is also a source of antibodies. Okay, nowadays the research is going on a cow colostrum for the treating diabetes. Okay, obviously, so that is what the antibodies are present in a colostrum, right? Then the next one is the artificial passive acquired immunity. So what is the artificial passive acquired immunity? So related with the artificial passive acquired immunity, that means if especially by natural way we are taking the antibodies in a by ready-made form, we are taking the antibodies from the another organism. So here we are using hyperimmunized horse or animals. Hyperimmunized horses, monkeys are used. Okay, hyperimmunized horse, from which the blood is taken out, okay, and just sort out the antibodies, and these antibodies are used to insert the another organism. So that is the artificial passive acquired immunity. So what is hyperimmunization? How we are making the horse hyperimmunized? So just during vaccination, what we are doing, we are injecting the weak or dead form of antigen into the horse. So obviously in response to that dead form of antigen, the body of the horse produces the antibodies. Okay, at very first time it is in a very less amount, but as the days progresses, first weaken, then quantity or percentage of that increases. Okay, and several times that horse is injected by the antigen. So what happens, every time that horse produces the antibodies. So nowadays, now that horse having a set of antibodies against that particular antigen, right? That means now that horse is hyperimmunized horse for that particular antigen. Okay, as nowadays we are knowing that the Hafkin as well as Serum Institute, okay, which are participating in the production of vaccine, they are using such a hyperimmunized animals to sort out the antibodies. So everyone, nowadays it is used. As you know, the plasma therapy. What is the feature? What is the characteristic or funda behind the plasma therapy? After 40 days, the person who previously having a COVID infection, suffered with a COVID infection, now its body has created antibodies against that COVID virus. So that antibodies are present in its plasma, right? So we are just sorting that plasma, taking that antibodies and injecting a patient who is just affected with the COVID. Okay, and this therapy is given. Okay, it must be matched properly. So especially this artificial passive acquired immunity is done against the during rabies. Okay, when dog bite happens to us, at that time we are taking the injections, that is the rabies one, or any animal's bite. Okay, even though the antivenom production, snake venom, okay, so against that snake venom, the antivenom is produced, that is also by such a way. Okay, so this is all related with the types of acquired immunity. Again, just look, it is of two types, active and passive. Active means our body itself producing the antibodies by natural way or by forcefully by artificial way. Then passive means we are just taking the antibodies in a ready-made form from the other individual. But naturally, we are gaining the antibodies from our mother when we are in a fetal condition. Via placenta, we are gaining that antibodies. While during the afterbirth, the colostrum is the richest source of the antibodies. Then the next one, artificial way. During artificial way, from the hyperimmunized animals who are taking that antibodies and injecting it into us, and that is the artificial way of passive acquired immunity. So this is all related with the types of acquired immunity. It can be asked for 2 marks. Okay. Then the next one is the cells of acquired immunity. Next point is the cells of acquired immunity. Cells of acquired immunity. Very important point. For short note, cells of acquired immunity, right? So, generally, there are especially the lymphocytes which are participating in a immune response, lymphocyte, and antigen present in cells. There are 2 cells. First one is a lymphocytes, and the second one is antigen presenting cells. Antigen presenting cells. These are the 2 major cells which are especially used for our immune response, right? So this, first of all, we are dealing with the lymphocytes. Lymphocytes are the cells of present in a lymph nodes. These are present in a lymph nodes. Okay, produced in a lymph node, so named as a lymphocytes. Okay, it is a type of WBCs. So, majorly there are 2 types of lymphocyte. Okay, there are 2 types of lymphocyte. One is produced in a lymph nodes, so named as a lymphocyte. Lymph nodes, everyone knows, na? You have studied circulation topic in that. There is a network of lymphatic system in our body. There are the lymph nodes, the pin-shaped structure which is scattered from our head to our legs, right? And these are majorly, the lymph nodes are present in majorly found in our armpits, groin region. Okay, and there are some lymphatic organs are also there, like tonsils, spleen. Okay, Peyer's patches are the example of lymphatic organs. So lymphatic organs as well as lymph nodes are responsible for production of lymphocytes. Okay, and lymphocyte is a type of WBC, as everyone knows, right? So there are 2 types of lymphocyte. One is a B lymphocyte and the another one is a T lymphocyte. First of all, we will study related with the T lymphocyte. T lymphocyte, and the another one is a B lymphocyte. So first of all, we will study related with the how the cells get formed. Generally, we have a hematopoietic stem cells which are present. For example, this is a hematopoietic stem cells. This is a hematopoietic stem cell. So as you know, the stem cells These are dedifferentiated cells, has an ability of dedifferentiated. That means dedifferentiated cells, to whom we are calling the cells which can form any type of any type of cell is called as a stem cells. Okay, so these are hematopoietic stem cells. So these hematopoietic stem cells are present in a red bone marrow, especially present in a red bone marrow. Suppose this is a bone, and this bone at the center side, it having a red bone marrow. This is a red bone marrow. Okay, the source of stem cells is a bone marrow. So this is a red bone marrow. So this red bone marrow having, yes, hematopoietic stem cells. It having a hematopoietic stem cells, right? Clear, everybody? Okay. So this hematopoietic stem cells during blood formation, that means hemopoiesis. Okay, formation of blood. Okay, that time it get divided into myeloid cells and lymphoid cells. It get divided into myeloid cells. Myeloid cells, and the another one is a lymphoid cells. Two types. Okay, it get divided into two myeloid, myeloid cells, and the another one is a lymphoid cells. Further on, these myeloid cells are forming our WBCs, as you know, neutrophil, eosinophil, then next one is a eosinophil, then basophil, then macrophage also. Macrophage. Okay, so number of cells especially are forming neutrophil, eosinophil, basophils, even though the mast cells and erythrocytes. Mast cells and erythrocytes. Mast cells and erythrocytes. Okay. So, as I have stated that the stem cells are dedifferentiated type of cell. Dedifferentiated type of cells means they can differentiate or they can form different type of cells, right? Are the sources to form a different type of cells, right? So, the next one is a lymphoid. So, related with the lymphoid as well as it also forms, the myeloid cell is also forming the dendritic cells. It is also forming the dendritic cells. Okay, so different type of cells get formed. Okay. Dendritic cells also get formed. Now from the lymphoid cells, from this lymphoid cells, three different type of cells get formed. One is B lymphocyte, then another one is a T lymphocyte, and NK cells, natural killer cells. Okay, so different type of cells get formed, and this all happens into the bone marrow. Clear, everyone? So, especially listen carefully. This is for your extra one. This is important for you. Can be asked for CET as well as LEAT. So by considering that, focus on this chart. Hematopoietic cells, hematopoietic stem cells which are present in a red bone marrow has an ability of dedifferentiated. Okay, during blood formation, it is majorly divided into myeloid cells and lymphoid cells. The myeloid cells are responsible for formation of WBCs as well as RBCs. Along with that, the mast cells are also formed. Okay, and dendritic cells are also get formed, right? Then the next one is the lymphoid cells. The lymphoid cells, especially, are forming B lymphocyte, T lymphocyte, and NK cells. Natural killer cells get formed. Now we are turning towards the lymphocyte, especially T, to whom we are calling as a T lymphocyte and a B lymphocyte. Okay, as you know, here from the red bone marrow, the lymphoid cells get formed. Lymphoid cells get formed. After that, these cells are divided into two. We are dealing with the B and T lymphocyte. Okay, the first one is the T lymphocyte. And the another one is the B lymphocyte. To whom we are calling as a T lymphocyte. As the T lymphocyte get formed, okay, this lymphoid cells, those lymphoid cells which moves towards the thymus. Okay, those which moves towards the thymus, those lymphoid cells which get mature into the thymus, okay, from red bone marrow, the lymphoid cells, some of the lymphoid cells goes to the thymus, get mature in the thymus, and here I will mention the lymphocyte. Okay, those lymphocytes from the red bone marrow goes to thymus, and they get mature into the thymus, and this mature lymphocyte from the thymus are called as a T lymphocyte. These are called as a T lymphocyte. Okay, now to whom we are calling as a B lymphocyte. Okay, during blood formation, the lymphocytes, those lymphocytes get synthesized into the bone marrow and get mature in a bone marrow itself. Okay, it's called as a B lymphocyte. So listen everyone carefully. To whom we are calling as a B lymphocyte, especially B is named because of the bursa fabricus. B is because of bursa fabricus. So this bursa fabricus, okay, this is the organ which is present in, this is the part which is present in a birds. This is present in a birds. But this part which is present in birds, but it is absent in human. But same function, okay, is performed in a human by the red bone marrow. Okay, so similar in birds and in human. Okay, in birds, bursa fabricus is there, and in human, red bone marrow. Okay. So, because of bursa fabricus, the lymphocytes are named as a B lymphocyte. Yeah, bursa fabricus is the part of the bird which is very close to the cloaca. Cloaca is the just end part of the alimentary canal of the bird. Okay, so it is a bursa fabricus, and this bursa, because of this bursa and because of this synonyms, the same organ, same function performed by the organ which is present in a human, that is a red bone marrow, and because of B for bursa and B for bone marrow, the lymphocyte which gets synthesized into the bone marrow and get mature into the bone marrow, these type of lymphocytes are called as a B lymphocyte. These are called as a B lymphocytes. Clear everyone? To whom we are calling as a B and T lymphocyte. Clear to everyone? Yes, is that clear? Okay, so this is all related with the types. Further subtypes of the lymphocyte we will study in a next lecture. Thank you.